Chapter number 2. Gene, Nature, Concept and Synthesis. Richard Dawkins in his book, The Selfish Gene, 1989, has stated about the gene, they are in you, and in me, they created us, body and minds, and their preservation is the ultimate rationale for our existence. They go by the name of genes, and we as their survival machines. The clear concept of gene came out after the work of T.H. Morgan, who gave the theory of genes for which he was awarded Nobel Prize in 1933. The recent work on structure of chromosomes and DNA clearly defines the gene as a small segment of polynucleotide chain consisting of hundreds or thousands of nucleotides. Nucleotides are the subunits of a gene. The total complement of genes in an organism is known as its genome. The genes are stored on one or more chromosomes and instruct the cells to make proteins. The size of genes in different organisms or within one organism that consist of more than one chromosome varies. In this chapter, physical and chemical nature of genes, classical and modern concepts of genes, their expression and regulation, and artificial synthesis have been discussed. A chemical nature of gene. The DNA is found in all plants, animals, prokaryotes, and some viruses. In eukaryotes, it is present inside the nucleus, chloroplast, and mitochondria, whereas in prokaryotes, it is dispersed in cytoplasm. In plants, animals, and some viruses, the genetic material is double-stranded, ds-DNA molecule, except some viruses such as +X174. In TMV, influenza virus, poliomyelitis virus, and bacteriophages, the genetic material is single-stranded, ssRNA molecule, table 2.1. The entire genetic message that controls the chemistry of every cell of the body acting in a specific way is actually written in the language of 4 nitrogenous bases of DNA, i.e. purines and pyrimidines. The defined sequence of the 4 bases constitutes a gene, which may be a few or several hundred base pairs long. Genes are the structures of the blueprints called protein which control the infinite variety of life. See Table 2.1. Nature of genetic materials. 1. Chemical composition. Purified DNA isolated from a variety of plants, animals, bacteria, and viruses has shown a complex form of polymeric compounds containing 4 monomers known as deoxyribonucleotide monomers or deoxyribotids, Fig. 2. L. Each deoxyribonucleotide consists of pentose sugar deoxyribose, a phosphate group, and a nitrogenous base, either purine or pyrimidine. Purines bases, adenine and guanine, are heterocyclic and 2-ringed bases, and the pyrimidines, thymine and cytosine, are 1-ringed bases. The following components of deoxyribonucleotide have been described. 1. A 5-carbon ring. Deoxyribose is a pentose sugar consisting of 5 carbon atoms. 4 carbon atoms, L2, 3, 4, of this sugar combine with 1 oxygen atom and form a ring. The 5th atom, 5, forms CH2 group which is present outside this ring. 3 O groups are attached at position 1, 3 feet and 5 feet, and the hydrogen atoms combine at position L2, 3 feet and 4 of carbon atoms, Fig. 2.1. In ribonucleotides, the pentose sugar is ribose, which is similar to deoxyribose except that there is an O group instead of H at 2 feet carbon atom. The absence of O group in DNA makes it chemically more stable than the RNA. 2. Nitrogenous base. There are 2 nitrogenous bases, purines and pyrimidines. The purines are double-ring compounds that consist of 5-membered imidazole ring with nitrogen at 1, 3, 7 and 9 feet position. The pyrimidines are single-ring compounds, the nitrogen being at position 1 and 3 feet in 6-membered benzene ring. A single base is attached to I carbon atom of pentose sugar by N-glycosidic bond. Purines are of 2 types, adenine A and guanine G, and pyrimidines are also of 2 types, thymine T and cytosine C. Uracil, U, is a third pyrimidine, Figure 2.2. A, G and C are common in both DNA and RNA. U is found only in RNA. 3. A phosphate group. In DNA, a phosphate group P043 is attached to the 3-carbon of deoxyribose sugar and 5-carbon of another sugar. Therefore, each strand contains 3 feet end and 5 feet end arranged in an alternate manner. Strong negative charges of nucleic acid are due to the presence of phosphate group. A nucleotide is a nucleoside phosphate which contains its bond to 3 feet and 5 feet carbon atoms of pentose sugar that is called phosphodiester. See Figure 2.2, Nitrogenous bases of nucleic acid. 2. Nucleosides and nucleotides. The nitrogenous bases combined with pentose sugar are called nucleosides. A nucleoside linked with phosphate forms a nucleotide, Fig. 2.1. Nucleoside equals pentose sugar plus nitrogenous base. Nucleotide equals nucleoside plus phosphate. On the basis of different nitrogenous bases, the deoxynucleotides are of following types. I. Adenine A equals DEOXYADENOSINE3, 5, monophosphate 3, 5, DAMPE, guanine G equals deoxyguanosine minus 5 feet monophosphate 5, DGMPE, thymine T equals deoxythymidine minus 5 feet monophosphate 5, DTMP IV, cytosine C equals deoxycytidine minus 5 feet monophosphate 5, DCMP. In addition to the presence of nucleosides in DNA helix, these are also present in nucleoplasm and cytoplasm in the form of deoxyribonucleotide phosphates, e.g., deoxyadenosine triphosphate (DTP), deoxyguanosine triphosphate (DGTP), deoxycytidine triphosphate (DCTP), deoxythymidine triphosphate (DTTP). The advantage of these 4 deoxyribonucleotide in triphosphate form is that the DNA polymerase acts only on triphosphates of nucleotides during DNA replication. Similarly, the ribonucleotides contain ribose sugar, nitrogenous bases and phosphate. Except sugar, the other components are similar. However, uracil U is found in RNA instead of thymine. Generally, RNA molecule is single-stranded besides some exceptions. 3. Polynucleotide. The nucleotides undergo the process of polymerization to form a long chain of polynucleotide. The nucleotides are designated by prefixing poly to each repeating unit such as poly A, polyadinylic acid, poly T, polythymidyllic acid, M, lide G, polyganidylic acid, plot C, polycetidic acid, and poly U, polyuridylic acid. The polynucleotides that consist of the same repeating unit are called homopolynucleotides such as poly A, poly T, poly G, poly C and poly U. 4. Chargaff equivalence rule. By 1948, a chemist Chagaf started using paper chromatography to analyze the base composition of DNA from a number of studies. In 1950, Chagaf discovered that in the DNA of different types of organisms, the total amount of purines is equal to the total amount of pyrimidines. I.e., the total number of A is equal to the total number of T, AT, and the total number of G is equal to the total number of C. It means that AT equals G, C, i.e. A plus C equals L. In the DNA molecules isolated from several organisms, regularity exists in the base composition. The DNA molecule of each species comprises of base composition which is not influenced either by environmental conditions or growth stages or age. The molar ratio i.e. A plus T, G, C, represents a characteristic composition of DNA of each species. However, in higher plants and animals, AT composition was found generally high and GC content low, whereas the DNA molecules isolated from lower plants and animals, and bacteria and viruses was generally rich in GC and poor in contents. Table 2.2. The two closely related species will have very similar molar % G plus C values and vice versa. Thus, the use of base composition has much significance in establishing relationship between two species and in taxonomy and of species. See Table 2.2. B. Physical nature of DNA. I. Watson and Cricks model of DNA. In 1953, J.D. Watson and F.H.C. Crick combined the physical and chemical data generated by earlier workers and proposed a double helix model for DNA molecule. This model is widely accepted. According to this model, the DNA molecule consists of two strands which are connected together by hydrogen bonds and helically twisted. Each step on one strand consists of a nucleotide of purine base which alternates with that of pyrimidine base. Thus, a strand of DNA molecule is a polymer of 4 nucleotides i.e. A, G, T, C. The two strands join together to form a double helix. Bases of two nucleotides bonds hydrogen bonds i.e. A combines with T by 2 hydrogen bonds A equals T and G combines with C by 3 hydrogen bonds GC. 2.3a. However, the sequence of bonding is such that for every ATGC on one strand there would be TACG on the other strand. Therefore, the two chains are complementary to each other. A sequence of nucleotides on one chain is the photocopy of sequence of nucleotides on the other chain. The two strands of double helix run in antiparallel direction, i.e. they have opposite polarity. In figure 2.3a, the left-hand strand has 5 feet plus 3 feet polarity, whereas the right hand has 3 feet, 5 feet polarity as compared to the first one. The polarity is due to the direction of phosphodiester linkage. The hydrogen bonds between the two strands are such that maintain a distance of 20 A. The double helix coils in right-hand direction, i.e. clockwise direction, and completes a turn at every 34 A distance, figure 2.3b. The turning of double helix results in the appearance of a deep and wide groove called major groove. The major groove is the site of bonding of specific protein. The distance between two strands forms a minor groove. One turn of double helix at every 34 A sugar phosphate nucleoside makes the backbone of double helix of DNA molecule, figure 2.3b. See the figures 2.3. Antiparallel orientation of the complementary chains A and Watson and Cricks of DNA double helix B. The DNA model also suggested a copying mechanism of the genetic material. DNA replication is the fundamental and unique event underlying growth and reproduction in all living organisms, ranging from the smallest viruses to the most complex of all creatures, including man. DNA replicates by semi-conservative mechanism, which was experimentally proved by Matthew Meselson and Frank W. Stalin in 1958. If changes occur in sequence or composition of base pairs of DNA, mutation takes place. Though the presence of adenine, guanine, thymine, and cytosine is a universal phenomenon, yet unusual bases in DNA molecule also occur. In some bacteriophages, 5'-hydroxymethylcytosine (HMC) replaces cytosine of the DNA molecule when methylation of adenine, guanine, and cytosine occurs. This results in changes of these bases. 2. Circular and superhelical DNA. Almost in all the prokaryotes and a few viruses, the DNA is organized in the form of closed circle. The two ends of the double helix get covalently sealed to form a closed circle. Thus, a closed circle contains two unbroken complementary strands. Sometimes one or more nicks or breaks may be present on one or both strands, for example, DNA of phage PM2 Pig 2.4a. Besides some exceptions, the covalently closed circles are twisted into superhelix or supercoils, figure 2.4b, and is associated with basic proteins but not with histones found complexed with all eukaryotic DNA. See the figure, bacterial structure of DNA. These histone-like proteins appear to help the organization of bacterial DNA into a coiled chromatin structure, with the result of nucleosome-like structure, folding and supercoiling of DNA, and association of DNA polymerase with nucleoids. These nucleoid-associated proteins include Hu proteins, IHF proteins Hi, Pha, and Fis. In archaeobacteria, e.g. archaea, the chromosomal DNA exists in protein-associated form. Histone-like proteins have been isolated from nucleoprotein complexes in Thermoplasma acidophilum and Halobacterium salinarum. Thus, the protein-associated DNA and nucleosome-like structures are defected in a variety of bacteria. If the helix coils clockwise from the axis, the coiling is termed as positive or right-handed coiling. In contrast, if the path of coiling is anticlockwise, the coil is called left-handed or negative coil. The two ends of a linear DNA helix can be joined to form each strand continuous. However, if one end rotates at 3600 with respect to the other to produce some unwinding of the double helix, the ends are joined, resulting in formation of a twisted circle in opposite sense, i.e. opposite to inwinding direction. Such twisted circle appears as 8, i.e. it has one node or crossing over point. If it is twisted at 7200 before joining, the resulting superhelix will contain two nodes, figure 2.4c. The enzyme topoisomerases alter the topological form, i.e. supercoiling of a circular DNA molecule. Type 1 topoisomerases, e.g. E. coli top 8, relax the negatively supercoiled DNA by breaking one of the phosphodiester bonds in ds-DNA, allowing the 3,O end to swivel around the 5-phosphori end, and then resealing the nicked phosphodiester backbone. Type 11 topoisomerases need energy to unwind the DNA molecules, resulting in the introduction of supercoils. One of type 2 isomerases, the DNA gyrase, is apparently responsible for the negatively supercoiled state of the bacterial chromosome. Supercoiling is essential for efficient replication and transcription of prokaryotic DNA. The bacterial chromosomes is believed to contain about 50 negatively supercoiled loops or domains. Each domain represents a separate topological unit, the boundaries of which may be defined by the sites on DNA that limit its rotation. 3. Organization of DNA in eukaryotic cell. In addition to organization of Dna in prokaryotes and low eukaryotes as discussed earlier, in eukaryotes the DNA helix is highly organized into the well-defined DNA-protein complex termed as nucleosomes. Among the proteins, the most prominent are the histones. The histones are small and basic proteins rich in amino acids such as lysine and or arginine. Almost in all eukaryotic cells there are 5 types of histones. Example: H1, H2A, H2B, H3, H4. 8 histone molecules, 2 each of H2A, 11B, 11 2 each of H2A, H2B, H3 and H4 from an octamer of about 11 nanometers long and 6.5-7 nanometers in diameter. DNA coils around the surface of ellipsoidal structure of histones 166 base pairs, about 7 quarters turns before proceeding on to the next and form a complex structure, the nucleosome, figure 2.5ab. Thus, a nucleosome is an octamer of 4 histone proteins complexed with DNA. See figure 2.5 structure of histone protein. The histones play an important role in determining eukaryotic chromosomes by determining the conformation known as chromatin. The nucleosomes are the repeating units of DNA organization which are often termed as beads. The DNA isolated from chromatin looks like string or beads. The 146 base pairs of DNA lie in the helical path and the histone DNA assembly is known as the nucleosome coparticle. The stretch of DNA between the nucleosome is known as linker, which varies in length from 14 to over 10 base pairs. The high is associated with the linker region and helps the folding of DNA into complex structure called chromatin fibers, which in turn gets coiled to form chromatin. As a result of maximum folding of DNA, chromatin becomes visible as chromosomes during cell division. See structure of ribonucleic acid RNA. The RNA is usually single-stranded except some viruses such as TMV, yellow mosaic virus, influenza virus, foot and mouth disease virus, reovirus, wound tumor virus, etc., which have dsDNA. The single strand of the RNA is folded either at certain regions or entirely to form hairpin-shaped structure. In the hairpin-shaped structures, the complementary bases are linked by hydrogen bonds which give stability to the molecules. However, no complementary bases are found in the unfolded region. The RNA does not possess equal purine-pyrimidine ratio, as it is found in the DNA. Like DNA, the RNA is also the polymer of 4 nucleotides, each one contains D-ribose, phosphoric acid and a nitrogenous base. The bases are 2 purines, A, G, and 2 pyrimidines, C, U. Thymine is not found in RNA. Pairing between bases occurs as AU and GC. If the RNA is involved in genetic mechanism, it is called genetic RNA as found in plant, animal and bacterial viruses. The DNA acts as genetic material and RNA follows the order of DNA. In such cells, the RNA does not have genetic role. Therefore, it is called non-genetic RNA. The non-genetic RNA is of 3 types: 1. Ribosomal RNA, rRNA; 2. Transfer RNA, Rna, or soluble RNA, Srna; and 3. Messenger RNA, mRNA, or template RNA. These 3 types of RNA differ from each other in structure, site of synthesis in eukaryotic cell and function. The mRNA and Rna are synthesized on DNA template, whereas rRNA is delivered from nuclear DNA. These RNAs are synthesized during different stages. During cleavage, most of mRNA is synthesized, whereas Rna is synthesized at the end of cleavage. Synthesis of rRNA occurs during gastrulation. The total population of rRNA is about 90% of all RNAs. Like DNA, the RNA is not self-replicating, but it has to depend on DNA. Therefore, replication of non-genetic RNA is known as DNA-dependent RNA replication. Moreover, the genetic RNA of viruses is self-replicating, i.e., it can form its own several replica copies. Differences between the DNA and RNA molecules are given in Table 2.3. Difference between RNA and DNA. D gene concept.